Good evening, everyone. So, I believe I'm audible. So, let's start. The topic for my presentation that I have been allotted is bacterial cell capsules. So, let's begin. According to this topic, will see bacterial cells release a kind of material. This material is known as glycocalyx material, and this material is amorphous, viscid in nature. So further we see this glycocalyx material is divided in two main types, one of which is a capsule and other is a slimy layer. So today we'll be mostly studying about bacterial capsule. So what is a bacterial capsule? When a bacteria releases a glycocalyx material which is well organized, properly organized, and which does not go off by washing off or rinsing by water. Such organized material, glycocalyx material released by a bacteria, is called as bacterial capsule, in simple words. And the other type of glycocalyx material is a slimy layer, in which bacterial cells release this material, but it is not well organized. The organization is not proper. So what happens is when we wash it by water, it gets rinsed off easily. That is the major difference between a bacterial capsule and a slimy layer. So further as we go ahead, we'll find out that this bacterial capsule is mostly composed of polysaccharides. Okay, the basic composition or most of frequently found composition of this bacterial capsule is a polysaccharide. So let's see a few examples of bacterial species and the composition of their bacterial capsule. So first we'll see is pneumococcus. These bacterial species show polysaccharide as their main major composition of the capsule. Then meningococcus, it also shows polysaccharides. Klebsiella pneumoniae, it also shows polysaccharides. Exceptional case we see Bacillus anthrax is a species of bacteria which will show polypeptide as the composition of their capsule. An amino acid in this polypeptide is glutamide. Then we see Streptococcus pyogenes. Some strains of this Streptococcus pyogenes have hyaluronic acid as a basic composition of their bacterial capsule. So we understood what is bacterial capsule, what kind of bacterial species have major composition of polysaccharide as their composition of the capsule. Now we'll go further in the part function of bacterial capsule. So there are a list of functions of bacterial capsule that has been listed, out of which first is protection from phagocytosis. So this bacterial capsule will help the bacterial cell to get a protection from phagocytosis. Phagocytosis is a process where phagocytes, white blood cells, will engulf. If this is a bacterial cell or an antigen, foreign material, phagocytes, white blood cells will engulf it, surround it from all sides, and destroy it and remove the dead particles. This is phagocytosis. So, this bacterial capsule will prevent the bacteria from the process of phagocytosis, and that's a protection. Second, we'll see is cell lysis prevention. Cell lysis is the breakdown of the cell. So, from break of the cell by osmotic pressure or any other kind of thing, it will help and protect from cell lysis because of this capsule. Third, we see prevent bacteria from bacteriophage. Fourth, protection from drying out. Fifth, it also shows virulence properties. Capsules of some bacteria are toxic, and when get ingested in a host body, it will show virulence. So that is one of the capsule's function we see. Further, we see it produces biofilms, about which the slimy layer that we just now studied about. So what are biofilms? When a bacteria, number of bacteria producing slimy layer, they come together and they collectively produce a slimy layer. This make a film on the surface. Such kind of film on the surface is called as biofilm. Okay. Further, the seventh point we'll see, it gives nutrition to the bacteria. Some bacteria present on the teeth will ferment the sugar. Due to this fermentation of sugar, other bacteria get nutrition. So this is one of the function. It gives nutrition to the bacteria. Example, if it is Streptococcus mutus. Eighth function is bacterial cell can also work as a vaccine. So how they will work as a vaccine? The outermost layer works as an antigen of the capsule and thus can trigger immune response in the host's body, forming antibodies and acquired immunity is obtained. So we can see that bacterial capsule can be used, some of the bacterial capsule can be used as a vaccine also, like pneumococcus, meningococcus. These are two examples of it. So these were the functions, some of the 8 functions I have listed here about bacterial capsule. So we have read up till now about what was bacterial capsule, then the composition of the bacterial capsule, then the functions we have studied, and now we'll go in the staining or demonstration of bacterial. There are 3 methods by which we can stain a bacterial capsule. First one is negative staining, second is amphibian capsular stain, and the third is serological testing. So these 3 types we can use to stain a bacterial capsule. So let's study about negative staining. India ink or Nigro stain is a stain that is used for negative stain. In amphibian capsular stain, polychrome methyl blue is the stain that is used to stain Bacillus anthrax. And the third part is a serological testing, in which 2 types are there: Quilling's reaction, and second is capsular antigen. Now in Quilling's reaction, what is done is antisera plus methylene blue together are used to stain a particular species of bacteria, which is Streptococcus pneumoniae. So how it happens is, when antisera and methylene blue is mixed together, the capsule of the Streptococcus pneumoniae is is swallowed off, and because of which it helps us to identify this particular bacterial species, that is Streptococcus pneumoniae. Now the capsular antigen, which is the second part of serological testing, in which what we find is latex agglutation reaction is done. Latex agglutination. Okay. Here what we do is the antibodies are coated with latex and tested against the bacterial capsule antigen of the bacteria. So understood? Latex coated antibodies tested against the bacterial capsule antigen, and then we find the agglutination reaction, which helps us to identify the bacterial capsule which it is. Examples of these are pneumococcus, meningococcus, and some fungi like cryptococcus, like this. So up till now, this was all about bacterial capsules. Few questions we can see if we have time. If we see what is a capsule in bacterial cell, it is organized glycocalyx material outside the cell produced by a bacteria which does not go off by washing. This is the answer. What is a slimy layer? Slimy layer is nothing but glycocalyx material which is not well organized, which goes off easily by washing, is slimy layer. Major constitution of capsule, polysaccharide. Major constitution of capsule in Bacillus anthrax, polypeptide. And what are the functions of capsule in bacteria? We can name 2. First, prevention of phagocytosis. Other, prevention from cell lysis. So this is an overview summary of the entire presentation. I hope you understood it, and I thank you for your valuable time. Thank you.
Thank you, Aditya.